Okay, questions, please.
So you say something about the pruning and like looking at the difference with the different tracks, you know, with the pruning, trying to see how this effect of getting rid of the unuseless connection. Wouldn't be, do you know what, with brain network analysis, wouldn't be like more looking at the connectivity would be more helpful to look at this effect of pruning, like with other, like looking at some other metrics of brain network analysis?
I've not actually looked at that level, at the network level. I don't know if you've looked at it just in the literature at all, Axel?
There is not much.
Not to say there isn't necessarily that much. I haven't looked at it in as much detail.
I think John Lewis has done it with diffusion data specifically, right? Because obviously with functional connectivity, people do it all the time. Yeah.
And they find something like about this.
You know, I mean, the problem is that it's often done in this kind of a blind way, voxel to voxel streamlining, which makes no sense at all, because you might find a streamline. I mean, if you look at the streamlines, possible streamlines using each voxel at the gray-white boundary, for example, and then see whether any streamlines reach any other voxel in cerebral cortex, you may come up with a lot of junk.
Right.
So I think the idea, so people who have done graph theory analyses using diffusion data, I always wonder a little bit, because it's completely data driven and ignores what is known about the major tract organization in the human brain. So
you don't trust the graph?
Well, I mean, you have to.
I think I'd have to know a lot about just what the tractography method is, because there's some that are better than others.
Right.
I mean, so it's not necessarily too blind, or if you can do something probabilistic.
Yeah, I mean, the idea that there's some connection from every voxel in cortex to every other voxel in cortex is, of course, completely ludicrous.
Yeah.
And there's no direct connections. There, of course, there might maybe polysynaptic connections, but yeah.
Yeah, in fact, they're debating what is the best way to go still.
But don't you have the same issues with functional connectivity matrices? Because you're kind of still testing every voxel to every voxel.
Well, but there's no assumption of monosynaptic connectivity in fMRI. So. But when you look at streamlines in diffusion imaging, you do say, if I find a streamline, that means there is some direct connection.
It depends on what approach you take.
Yeah.
I mean, it's, again, you can do, like what we do with FSL is streamlined, but it's probabilistic. So it's like, there might be.
Undeterministic.
Right.
It's hard to interpret the group differences in these tracks, right? So how does the behavioral data help us interpret, like, have people found, like, sensory is associated with these differences in these tracks, or is it more that it's kind of a general difference that's hard to sort out?
No, if you do correlations, you can find as you go, you can find correlations between the developing tracks and some of the specific cognitive functions. I'm sorry that I didn't get all that in here, but yeah, that you do find correlations.
Well, there's also some recent studies on presumably they find correlation with changing behavior within very short period of time, like 24 hours.
Yeah,
there's behavior. Some learning has occurred.
Right, they'll do a training process. Yeah, you can find, which always surprises me,
I know.
but that you can see it. You can actually see like changes in the FA levels of a tract as you've done. Like, I think they've done piano playing and things like that are the ones that I've looked at, but there's a lot more nowadays of interventions or training or whatever.
Juggling, I think they've done juggling.
What's that?
I think they've done juggling.
Have they done juggling too?
Early ones, yeah.
Yeah. So various ones like
can the students in class ask some questions? They are supposed to have some questions prepared, and don't hesitate to ask really basic questions. Those are always the most difficult ones.
Oh, no, I was those are easy.
I
We'll
was just
see.
curious,
We'll see,
I know
yeah.
you didn't really have time to talk about the later portion of the lifespan, but if you had any particularly interesting sources using these methods for like the aging brain or degenerative diseases.
Sources, not immediately, but I did actually give us a few slides anyway, if we wanted them, once I find them again, because they're down here at the end. Oh yeah, here we go.
Wow, the hippocampus.
I know.
You don't want to go to 100.
Yeah, not as well labeled on these. So I took some very narrow ones and stretched them out to make it a little easier to see. Looking here, we're looking at 20 up to 90 something. And yeah, what you see with the different structures is kind of what you see with a lot of aspects of aging. Lots of aspects decline at a fairly steady rate. Some will then start to speed up about 50 or 60 years of age. This is the normal hippocampus.
This is volume?
This
Total
is-
volume of it?
Oh, this one, yes, I'm sorry, this is volume. I think I had, did I have some for diffusion? No, I didn't have anything for diffusion. Yeah, see, when you leave it this narrow, it's just kind of terrifying.
Yeah.
It's like cerebral cortex. But interestingly, it's like here, look at cerebral white matter, nice and steady, and then it goes down much more rapidly. I think that's partly from hyperintensities and things. So there's other pathologies that you start to see with aging, particularly around the ventricles. Where the contrast starts to change. I think it's partly myelin, but I'm not actually sure what all the contributors are. But I think that's part of why you get this like elbow in cerebral white matter volume. Hippocampus doing about the same thing at 60 or 70, but then a lot of the other structures just showing a nice steady decline. But then of course for Alzheimer's or something like that, you see much steeper declines.
I was thinking about with the little bits that you were showing us with individuals with autism and the kind of accelerated development in some way in like particular areas, or how they just kind of had higher volume in those areas earlier on, I think it was, and I was thinking how the boys were just larger in general, and there's a higher incidence of autism in males, and I'm wondering if that's related or if we know anything about that.
Simon Baron-Cohen
Simon
loved
Baron-Cohen.
that question.
One of the theories, particularly there's a guy in the UK, Simon Baron-Cohen. One of his big theories is that he describes autism as the overly male brain. That maybe it's the brain going more so in one direction. That doesn't fully fit with like what we see. So yeah, we saw increased volume in autism, but we also see an earlier development. So, and I say development, but really it's increases in volume measures. I don't want to say it's, true, healthy, successful development, because there's other things going on. But it's, it's happening much earlier, and that seems to be a key aspect of it. And in that sense, if we want to say it's earlier, well, maybe that's an overly female brain at the same time, because girls are usually earlier, right? But that is one of the things that's been thought about. A lot of developmental disorders, though, have higher rates in males. So quite a lot of, I think, ADHD, somebody else name a few. I'm not sure. There's several, I believe, though, that have higher rates in males. Pardon me?
Language impairment.
Language impairment, yes. So there's quite a few that are more common in males. So y'all are just less resilient, apparently.
That's why there's so few of us here.
That's why there's so few. You just can't take it.
I have a related question. So for the autism, they have the more volume of gray matter, right? And I think you said, but could you go into a little bit more detail about how that changes over time?
Yeah, and so like I said, what we see I mean, it took a while to discover that in the research because, you know, the earliest studies of autism, I'm trying to get to that one figure there. Earliest studies of autism were looking at adolescents and teenagers. They're a lot easier to find, or would look at adults more likely, where you don't actually see a difference. If you look at in these older age ranges, you don't usually see a group difference, at least up into early middle age, maybe up into the 30s or so. You see it mostly just before the, say, 5 or 6 years of age, where again, what you're seeing is not that autism is declining in these measures, but that the typical group is catching up. So what you have is the rate of growth is accelerated on autism in the first years, and we think a lot of it's actually postnatal. Accelerated in those first years, but then is abnormally slow for a period as the control group catches up. And then they may be fairly comparable. And then what we're trying to look at as we start looking at aging is whether or not there's a decline later on. You know, I didn't get to aging, but one of the things that you see in, as you look neuroanatomically, is sort of the last in, first out concept, for one thing. With the idea that the areas of the brain that develop last seem to be the most susceptible to decline later on. Maybe also with that, in autism, we've got abnormal development. Maybe those, the aspects that are atypical are also less resilient. Maybe, you know, and again, like frontal and temporal lobes, we know seem to be the most abnormal with regard to the early volume. Maybe that means they've got an extra susceptibility on top of what they'd already have for normal aging. There's still not a whole lot of studies out there, but there may be some suggestions that say executive function might have issues even earlier in autism. I don't know that yet, but there's, I think I saw a poster, certainly when that comes to you. And I'm wondering whether or not maybe motor function might be particularly susceptible. But really nobody's looked beyond about 50 years of age to see what happens in later age.
Okay, thanks.
Do we have any more questions from the class?
Mine's completely unrelated, and it's very basic methodological question about DTI. I think in the article that we read, it was the sample size was like 200 something. Is that necessary for DTI, or is there like a lot of variability, or?
Not for DTI, but for autism.
Okay.
So, right, so DTI can be valid, but there's so much variability in autism and the brain structure in autism, and we think there's subtypes, and there's individual variability. I won't look for it in here, but there's so much variability you have to get around that. So 200 is a big study, at least for, say, a single lab, but it's not uncommon to see much smaller than that, certainly. Say 30 or 40 a group is nice. I mean, that, or is common in the literature. How's that? Depending on how much of an age range you want to look at, you'll need more. Some of the typical development ones that I showed you are, say, 1,000, I said. That's obviously multi-site things, though. So like the Ping study that I showed you, which is a very nice samples of typical development, have like 5 or 6 different universities they're working with. And with autism, we've now got some networks where we're collecting imaging data so that we can start to get at that, the variability that we get between individuals.
More questions from the students in class?
I had a question. If a person is having an Alzheimer's disease, so how
Sorry, is that
Alzheimer's?
Yeah.
So does the diffusion direction really changes, like with the? I mean, how do you detect it with this imaging scheme that a person is having Alzheimer's?
I'm not actually studied up on Alzheimer's in particular, so I don't know that. But you can actually see, say, looking at typical aging, you can see changes in all sorts of the measures. You're not likely to see changes in the anatomy itself.
Yeah.
So I wouldn't expect to see, say, changes in the direction of diffusion so much.
I read
But-
it somewhere that since there is a change in the cell orientations, like it said that some cells degenerate, like in the areas majorly near hippocampus. So I read it somewhere like the direction of diffusion is also like it's a measure that you can detect if a person is affected with Alzheimer's or not.
So I guess two things. One is what's going to happen is at least as axons degenerate, you're going to have things like, okay, say if you lose myelin, then your radial diffusivity can go up. And so what you're really going to get is like an increase in the variance of the direction. Because if you have a nice, healthy bundle of axons, it's going to look like it all goes this way. As it starts to loosen up, it's going to be sort of like lower confidence of the direction in a way, and more variability. And you can also, if you look specifically at some of the submeasures, so I didn't say a whole lot about axial diffusivity and radial diffusivity, but you may be, depending on, of your 4 different measures, which combinations are, say, deteriorating, you might be able to get some information on, well, is it the cell itself that's having problems, which may have more of an effect on the axial diffusivity? Is it the myelin, which may have more of an effect on the radial diffusivity? You might be able to detect that the axon is deteriorating, but the myelin's still okay. So there's lots of things that can contribute. If you looked in an area, if you have the resolution to look in an area where you have very nicely oriented cells, like say in certain areas of the hippocampus, and you need good resolution, maybe you could see something more with regard to the orientation if you're, say, losing, well, yeah, I wouldn't expect it to, like, you know, change the direction really, because that would be almost like the axons are tipping in a certain sense. But you do get a loosening and then a lot more variance in what those measures are. And if you've got something, like I said, like the crossing fibers, if you lose one bundle, that'll change the direction. That'll totally change the direction.
So for autism, again, is there any changes in like the white matter development, or is it just the gray matter that's kind of peaking earlier?
So we're seeing, I was trying to think, oh, why did I have that in there? Yeah, we see white matter differences. So we see increased fractional anisotropy. We see it with an earlier development, just like we do with gray matter. Volumes of white matter, if we look at it not with diffusion but with anatomical, we also see an increase in the volume of white matter early on. And it may be more so in some of the areas where you see the bigger bundles, more so there than in areas that have a little bit less. But we do see white matter differences, which is not surprising because, you know, that's the axons coming out of the neuron. So if you saw one but not the other, that would be kind of strange almost.
So looking at this graph, you see that you see the change starting from age of 2. Do you have some prenatal evidences to predict autism in kids, the prospect topic that you talked about?
Not prenatally. We don't know yet what exactly the causes are. We don't know genetics. A lot of the brain growth differences, like I say, may be postnatal. But the most important reason that we don't have something where we could predict is that you do have so much individual variability, both in the typical brain and in autism sort of has extra variability. So that all of these things can be very informative about what a group is like, but it's not going to lead you to, but in autism at least, we don't have differences that are so stark that you would be able to make a diagnosis with it. So it's not like a lesion where it's like a bright spot that you just don't see in other people. It's subtle differences, but when you look at a large sample, you can see that the groups differ on average. And so that can then lead you to possibilities of the explanation that you can follow up on.
Okay, we're a little bit over time, so thanks again, Ruth. And if I may ask people who are not in class, if you have questions for the lecturer, please to do it outside because we have a little bit of a wrap-up session here in the class. Thank you very much for coming.
That means I leave too. So that means I leave too.
Unless you